We should start.
Yeah.
Good evening, everyone. My name is Khushbu, Marketing Executive at Strand Life Sciences. And on behalf of the entire Strand team, it is my pleasure to welcome all of you to today's session. Strand Life Sciences is a genomics research and diagnostics company and a part of Reliance Industries Limited. Founded at the Indian Institute of Science, IISc, Strand has over 25 years of experience in transforming complex genomic data into simple, actionable insights, driving advancements in clinical diagnostics, translational medicine, and personalized healthcare. Today's webinar focuses on a highly exciting and rapidly evolving area: applications of single cell RNA sequencing in cancer biology. It is now my privilege to introduce Dr. Pankaj Kumar, Senior Bioinformatics Engineer at Strand Life Sciences, who leads a team developing cutting edge single cell and spatial transcriptomics pipelines. I would like to invite Dr. Pankaj to briefly introduce our speakers and take the session forward.
Thank you. Thank you, Kushbu. Hello, everyone. So, welcome to the seminar. And I first of all thanks all the speakers who generally accepted our invitations to participate in this webinar.
No, sorry.
Yeah. So, first of all, I would like to introduce Dr. Kamal. Dr. Kamal is has just a minute. Let me. Okay. Yeah. So the Dr. Kamal has did his master's in biotechnology from the NIT Durgapur, followed by the PhD from National Institute of Immunology in 2018, and currently he is holding as an assistant professor at the Gujarat Biotechnology University. In his professional career, he spent more time in the different US laboratories, which is more centered to the different aspects of the CAR-T and other biological, to address the other biological questions. Our other speaker is Dr. Rohini Nair. She is from the prestigious Banaras University, from where she did her PhD in 2015, followed by a very prestigious Marie Curie Fellowship and the postdoctoral experiences from the different European laboratories. So she will be presenting her work. And then our third speaker is Dr. Tanveer Ahmed. She did a master's in biochemistry from the University of Kashmir and then PhD from the CSIR Institute of Genomics and Integrative Biology, which is also the degree was from the Academy of Scientific and Innovative Research. And he's actively working as an assistant professor at the Multidisciplinary Center at Jamia Millia Islamia University. Then from our end, we have a Dr. Arifu, who is leading the experimental team of the entire single cell spatial and other commercial research projects. She did her PhD from IBAB in Bangalore, and then years of experience in the experimental biology from the various USA laboratories. She's here since, I think, 3 to 4 years, more than 4 years at a Strand, and handling the many projects, almost all the commercial projects related to the single cell and the spatial, as well as the other biological questions. I had did my PhD from the same institute, Institute of Genomics and Integrative Biology, IGIB New Delhi. And after that, I spent almost 7 years as my postdoctoral career, including from the and in STEM, NCBS, then the Karolinska Institute, as well as from the ULB University of Brussels. And here, since last 3 years, I am basically leading the team of the researchers who's working on to providing solutions in terms of the single cell and the spatial transcriptomics. So for this, I am now handing to Dr. Tanveer to talk about his work. Thank you. Over to, Tanveer.
Yes, thank you so much, Pankaj, for this kind introduction. So my slides are visible, I think.
Do it.
Excellent. So there was a missing piece of information, Pankaj. Since I completed PhD at IGIB, I moved to US, NIH, where I did my postdoc. From 2013 to 2017. My postdoc was on CAR T cells. We were making receptors, engineered receptors, chimeric antigen receptors. And I was also actively involved in the group who made the first ever CAR T, which got approved in US by FDA in 2017. And then in 2017, late November, I came back to India and joined Jamia Millia Islamia as assistant professor. Currently, I am on sabbatical. I am working with as a co-founder and director of Cellogen Therapeutics, where we are making next generation CAR T cells. I'm also actively involved with MicroCRISPR Private Limited, which is a Gujarat-based, Vapi-based biotechnology cell and gene therapy company, where I have been consulting since last 5 years. So thank you so much for the invitation, Pankaj. I'll be talking about CAR T cells, two cutting-edge technologies, CAR T cells and then single cell RNA-seq, and how these have recently changed the landscape of cancer detection and then the therapeutics of cancer. Particularly, how now we have a data of more than 1 billion single cell RNA-seq data of over 1 billion CAR T cells, and what we have learned from these single cell data, and how this particular data is going to help us to develop next generation therapies which are cell and gene based. So, taking back to when we started, it was 2012 when the first patient was ever treated with a second generation CAR T cell therapy. In this video, you can see Stephen Group is talking about Emily Whitehead, who was by that time around 10, 11 years old when she received the CAR T cell therapy. First ever pediatric patient to be treated with CAR T cell. I'll talk more about the generations of the CAR T cells. She received CD19 monospecific second generation CAR T, and as we speak, it has been almost 14 years. She is cancer free. That is the potential of these therapies. It is called a living drug. You need to give it one time, and this is Emily Whitehead now. Many patients now, we have data from more than 70,000 patients across globe, which shows the potential of this permanent kind of cure for the B cell malignancies. So the best part of the CAR T cell is when the first line defense fails, second line defense fails, third line defense fails, or fourth line, which is a immunotherapy. Even the patient, when they relapse with all the prior 4 lines or 5 lines of treatments, then CAR T cell was given, as in the case of Emily Whitehead. She was, doctors had given 3 months time for her to survive, and that is how now where she is after receiving the treatment. So after all the prior lines of treatment fails, then CAR T is something which is actually helping patients to live a longer and healthier life. So about briefly about the technology, as you can see in this brief video, when the normal T cells fail to attack cancer cells, what we do is we take the blood from the patient, we bring this blood to the clinical lab, GMP lab, where we isolate CD4, CD8 or CD3 cells. Then these cells are engineered with a lentiviral vector. Most of the cases, this lentiviral vector has a transgene which is called as CAR. I'll come to that slide next. And then these engineered CAR T cells are expanded in the lab and taken back to the clinic, infused into the patients, and then they actually bind to the cancer cells and eliminate them in the process. The process itself takes around 7 to 12 days. It depends on the manufacturing platforms which different companies and academic institutes use. So on an average, it is around 7 to 12 days from the day you take out the blood from the patient till the day you infuse back. The process is called as autologous CAR T cell therapy, and it is a kind of person specific treatment. So what makes these CARs unique is the binder, which is a chimeric antigen receptor molecule. So we have seen different generations of these molecules. First generation CARs were made in early 1990, and then second generation CARs were made in 2004-5. First generation CAR were tested in clinical trials, but they didn't show the optimal effect. Then second generation CARs is what we saw the tremendous efficacy, both as an example in pediatrics like Emily Whitehead and so many other patients who have lymphoma or leukemia, both pediatric as well as adults. So this is where most of the study research has gone in last almost 15 years now. And with what we have learned through single cell transcriptomics and other molecular assays, is that there is a need to come up with next generation molecules which have a better efficacy. I'll come to that slide also, why we need to come up with newer generation of cards. So the difference between first, second, third, fourth, fifth generation cards is, so most of the CARs for B cell malignancies, leukemia and lymphoma, have this binder which is CD19. So monospecific CD19 molecule which binds and identifies a cancer cell. Then on the T cells, when this molecule activates the signaling, first generation had only CD3 zeta. Second generation had one additional domain which is a co-stimulatory domain, either 41BB or CD28. 41BB is the first FDA approved product, which is Kimria by UPain, and CD28 was again same year approved by FDA, that is Yescarta, which has a CD28 as co-stimulatory domains. Then third generation have two co-stimulatory domains. Fourth generation also have a cytokines secreted by these CAR T cells to increase their persistence. So what we have done with, as I was giving example of microCRISPR, uh, my group has actively worked with them. We have generated the first ever third generation CAR T molecule of the country. This molecule was taken forward for the clinical trials. As we speak, we must, I think the data for phase 2 must have also been analyzed. This was phase 1 was completed last year, and now phase 2 has also been completed, and the response is tremendous. Almost 80% of response rate in leukemia, and lymphoma is slightly lesser. So but data is again not of many years, it's just 1.5 year data. So we will wait for the other data to be coming, and Dr. Dinesh Pendarkar, who is the PI of the study, he'll be talking more in different platforms about the clinical aspects of this. So what we have learned in these 10 years, particularly as we speak about single cell RNA-seq data, this is one meta-analysis which has come in Nature in 2024. So although in initial 1 or 2 year, the CAR T cells do show a tremendous response, but over time, particularly after 5 years, there is a significant decline in overall response rate and the event-free survival. Reason being two, what we have learned is one, they have a monospecific CAR T cells, which we are talking about at this point in time, and this meta-analysis has done is it's a CD19 directed monospecific CAR T. Second, they are most of the data is from a second generation CAR T. So we do not have much data from third generation or bispecific or trispecific CAR T cells. And what has happened is, although the initial response is nice, but over time you see the people who relapse with cancer, they have two major issues. One is lack of persistence. So these CAR T cells do not stay in the system for a longer period of time. That means every patient is not like Emily Whitehead. Almost 50% of the patients do relapse. Lack of persistence, the cells will not be there in the system for a longer period of time. Second is antigen escape. As I am talking about CD19 negative relapse, the patient will have now cancer which will not have CD19 on the surface, and even if the CAR T cells are there, they will not be able to eliminate that cancer. It's a kind of antimicrobial resistance pathway. So first part I'll talk about briefly lack of persistence and how we are addressing this lack of persistence CAR T cell issue, and it has been over a decade where my lab is actively involved in this. So single cell has actually transformed the way we look at CAR T cell therapy. As I was telling, more than 1 billion CAR T cell data is available. So with single cell data from the patients who have received these infusion products, before we infuse the CAR T cells to the patient and after we infuse it, so we take the cells at both the levels, and then we look at the patient who have shown the response and the patient who have relapsed. What are the molecular differences when we look at the single cell data? A lot many things we have learned. This is one example which came in Science Advances 2022. They have shown that 12 patient infusion products, they have pre-infusion products they have taken, and they did single cell RNA-seq, and what they found was very interesting. So the patients who did respond, they have a high level of cytokine, particularly Th2 cytokines, as you can see on the right side, BD. So there was an increase in Th2 type of cells, IL-4 was increased, and other Th2 cytokines were high in the responders. But the patients who did not respond, they have a very low levels of these Th2 cytokines. So that shows now the next generation CAR Ts which people are making, they are taking care of the Th2 responses. AI platform is now being integrated with single cells. Our lab is also actively working on AI platforms to make these next generation CAR T cells using single cell omics. These are some of the available single cell RNA-seq platforms which help us to design newer generation of CAR T cells and also address the molecular challenge with CAR T cells, like single cell GPT is being actively used. And then these models, we also have developed a couple of them. I think I'll not be talking about at this point in time about those AI models, but maybe next. So what we have done in these recent years, this is one example from my lab where we have used developed these fourth generation chimeric antigen receptor CAR T cells. These are the scientists who have been actually involved from AI domain, from computational domain, in silico bioinformatics, and then wet lab and preclinical studies. Has led the preclinical study, and Dr. Nisha, she has done the AI analysis of this. So we did same mice model. We isolated the CAR T cells from the animals, and then we did bulk seq RNA sequences. We also compared this with the single cell RNA sequences to find what are the changes which are happening in the CAR T cells when you expose them to the multiple tumor antigens. What was interesting in this analysis was when we found that there was a higher subset of memory CD4 and CD8 T cells which showed increase in autophagy process. And now if you increase the memory CD4 and CD8 cells, the chances of getting higher persistence of these cells increases. And when we did this, we also then did a molecular screening. We created a library of FDA approved molecules to find out which molecules are actually activating the or increasing the number of memory CD4 and CD8 cells. And we found a combination of semaglutide, which is a well-known weight loss drug, and urolithin, which actually increase the mitochondrial metabolism and biogenesis. These two combinations finally eventually increase the memory subset of these cells. We tested these molecules in multiple leukemia and lymphoma models, and we saw, as compared to the conventional second generation CD19 CAR T cells, these metabolically reprogrammed CAR T cells persisted much longer period of time. And one video example is here. This is a second generation conventional CAR T cell, where you see over the time these red are the cancer lymphoma cells. So on the right you can see this is a GLP CAR T cell which is optimized. The gray are the CAR T cells surrounded by it. You can see the number of these CAR T cells is high, and eventually all the cancer cells are eliminated in the culture. Again, we developed tumor rechallenge models, mice models, and we saw that these fourth generation reprogrammed armored CAR T cells performed better as compared to the second. This study was published in Cell Reports Medicine last year, and we have filed a patent in India, and US patent is currently pending. Coming quickly back to the second, Pankaj, do I have a couple of minutes? 3, 4?
Yeah.
Thank you. So coming back to the second challenge, which is currently in the field, antigen escape. As I was telling, most of the CAR T cells, which we also have now in India, they are CD19 directed, monospecific. And now relapses, which we see is there are CD19 negative relapses. So one example is again single cell data from patients, which they have taken 84 patients, and then they have all the pre-infusion products, CAR T cells they analyze, and they wanted to see what are the patterns. As you can see in this example also, almost 27 patients relapse with CD19 negative. And now we also have done meta-analysis to find out how many patients relapse with CD19 negative. Almost 30 to 40% of the patients relapse with CD19 negative. This is one example where you have a CD19 CAR. And how to overcome this? You need to target two different antigens. CD20 and 22 are the other antigens which are present, expressed by these leukemia and lymphoma cells. What we have done is we have made a bispecific CD19-20 CAR, and that is I'll be talking about. So that's a bispecific CD1920 fourth generation CAR T cells. So we have created a library of these CAR molecules. I think that is the largest library in the world, where we had screened almost 10,000 CAR molecules. Among those, 1,500 have been synthesized in vitro. Varnit, who is the computational biologist, he did all the analysis to find out the best combination of these, because the car interaction is very well known in this case, which leads to tonic signaling. So we wanted to make sure that these cars do not self interact or aggregate, so that they can have their desired activity. Then Nisha, she went forward, she developed this CalmSat model. This paper has been published last week in Nature Communications, where we have screened these molecules and tested. We also did the membrane docking of all these CAR molecules, 10,000 molecules, and to find out the best binder combination. And finally, the binder which showed the optimal result was taken forward. Arij, she did all the in vitro assays. As you can see on the right side, these are the leukemia cells, where you use this bispecific CD1920 CAR T, and you can see how these leukemia cells are eliminated over time. This is another example of a live cell, where you see the lymphoma, red lymphoma Raji expressing cells here, and surrounded by CAR T cells. Over time, you see how these lymphoma cells are eliminated from the system with this bispecific. So this is the bursting of the lymphoma which you see. We have tested in multiple animal models. Again, toxicity study was done, GMP batches were made, and now this has been. This is another example where you see the mice which are here. They are the tumor mice. They get paralyzed and they are not able to move. We then inject these mice with the CAR T cells through tailwind injection, and then you can see these are the mice which have received the CAR T cells. Then now they are happy and healthy without any tumor-related symptoms. We also have patented this particular technology. In India, we got this patent approved. We have filed global patents in 14 different countries. US patent is expected to be by this year end approved. We have questions and we have addressed those queries from them. So we also have filed a patent for the lentiviral vectors, which goes into making these CAR T cells. As I was saying, these are the studies which are already published. They are available in the public domain. Particularly AI guided car design, which came in Nature Communication last week, that is available, where we have extensively used single cell, bulk RNA, and then AI guided. So this is the team. I genuinely thank, and I am always thankful of Dr. Anurag Agrawal, with whom I started my journey of cell and gene therapy in 2008 when I joined his lab, where we were using lentiviral technology for mesenchymal stem cell regeneration. And then Dr. Gaurav Kharia, he is co-founder and director at Cellogen Therapeutics. Dr. Shiva Prakash, he is associate professor at IIT Kanpur and the scientific advisor with us at Cellogens. And the team, smart team who's here. And all the clinicians, Dr. Vikram Matthews, he's helping us with the phase 1 clinical trial for the bispecific CD19 CAR T at CMC Vellore. Clinical trial application has been submitted for that, and hopefully we'll be starting the trial in another couple of months for that next generation bispecific CD19 CAR T molecule. And these are the lab researchers who are the backbone behind this particular study, these two studies which I have presented. Yes, thank you so much.
Thank you. Thank you so much, Tanvir. Very nice piece of work. So, Kushbu, we will take the questions in parallel or in the last?
We can take the questions at the end, sir.
Okay. Yeah. So, the next speaker is Dr. Kamal Mandal. So, I'd like to invite Kamal Mandal to yeah.
Yeah, first of all, thank you, Dr. Pankaj, for the kind invitation and the fantastic talk by Dr. Tanbir Ahmed. So I think he already has set the tone and the foundations to the audience, like what is CAR T therapy and how is it actually helpful and how powerful is it. So just in continuation to that, I'd say that the CAR T therapies conventionally we target the cell surface antigens. And so basically we target the antigens which are present on the surface of the cancer cells and not on the normal cells. So that's how we we can discriminate between the cancer cells and specifically kill only the cancer cells, leaving the cancer cells, normal cells untouched. So as we all know that the cancer markers, like they're present exclusively on the cell surface of cancer cells, are very rare, and like there is an acute dearth of that. So we went a few steps ahead and thought, like, can we find some conformationally selective cell surface cancer antigens, like same protein in the cancer cells and the normal cells, but in the cancer cells they are conformationally different, and we can target the conformationally selective cell surface antigens of the cancer. So that was the idea. So that's what this whole talk is going to be about. Uh, just having trouble the slide. Yeah. All right. So, immunotherapy has emerged as a very powerful tool for cancer care. So broadly, we can say like there is antibody therapy and CAR T cell therapy. And what we do actually in CAR T therapy is that we basically take the variable heavy and the variable light chain of antibodies, and that we engineer and make it express on the surface of the T cells. And now the T cells can directly recognize the tumor cells through this antibody fragment, and the variable heavy and the variable light chain of the antibody fragment. That's what is responsible for binding or specifically recognizing to the cancer antigens. So when we make it express on the surface of the T cells, that now this T cells is very powerful and can bind to the tumor cells more specifically, and that's what leads to specific killing of the T cells. So this technology is very matured by this time, but the clinical applications is still limited to B cell malignancies, I would say. Like it has not expanded beyond that. Like there's some approvals for multiple myeloma, but the multiple myeloma cancers are also, it's derived from B cells. As it starts from B cells as precursor. So it has like there are like no clinical approvals beyond B cell malignancies at this point, I would say. So there's and one big reason behind this is that there's acute dearth of cell surface antigens, which are exclusively present on the cancer cells. So that's what we worked on. Like this is a challenge that we wanted to work on. And cell surface proteins are very attractive in the sense that they are pharmaceutically very often targeted. So most of the pharmaceutical drugs, like 70% of them are targeted to the cell surface proteins. And cell surface protein expression does not correlate very well with the transcript level. Like if you do RNA sequencing, it would not correlate with the cell surface protein levels. So one way to do it, like to see the cell surface proteins, is that we can just label them with something like biotin. And what we do is that the cell surface proteins are heavily glycosylated, so we just install a biotin on the cell surface proteins used by chemically modifying or basically glycooxidation of the glycosylated cell surface proteins using sodium metaperiodate. And then once we have biotin on the cell surface proteins, we can just go for a pull down with the streptavidin beads and do mass spectrometry. And that's how we identify the cell surface proteins. And this technology we have extensively used uh, to identify new cell surface targets, the surface of the cancer cells. And we have done it in the past, that in the context of B cell malignancies, like Tanveer explained very well, that we target CD19, but a lot of time when this antigen escape, we go for CD20, CD22, those are on the queue of clinical trials and approvals. But there is still acute dearth to address the acute dearth of this cell surface markers of these cancer cells. So we did this cell surface proteomics, where we enriched the cell surface proteins with this chemical biology strategy followed by mass spectrometry. And then we identify CD72 as a specific target of B cell malignancy. And then we use this East display platform to engineer a nanobody. Nanobodies are nothing but it's also like antibodies of camels. So unlike humans, where we have heavy chain and light chain, camels have only light heavy chain. And because of this heavy chain, it becomes easy to engineer nanobodies against our desired antigen. So we did that. So we engineered a nanobody against CD72, and then we engineered this nano CAR T against B cell malignancy as an alternate to the existing CAR T therapies targeting CD19. So the CD72 CAR T can be used as an alternate in case when the relapse from CD19 CAR T treatment. So even with all these, we still believe that there's the dearth of cancer-specific cell surface marker that's still not addressed, and we thought that maybe we should go a few steps ahead and find conformationally selective, uh, uh, selective cancer antigens. So, but there was no technology to find this kind of cancer antigens. So what we believe is that maybe the same set of proteins are present in the normal cell, the cancer cells, but in cancer cells, they look conformationally different. And with that, we can engineer some antibodies which specifically bind to this exposed epitope of this conformationally different antigen, and that will spare the normal cells and bind only to the cancer cells. And once we have that kind of antibody, we can derive a CAR T out of it using that antibody or nanobody, and then that's how we'd be able to target the cancer cells exclusively. So one way to do so that we thought that we developed using cross-linking mass spectrometry, it's possible to probe the rough structural conformations of these proteins at a coarse level. It's not like a high resolution structure solutions like we get in crystallography. So it's a coarse, like we can identify, like you get a rough idea of how the protein looks like in 3D space while it's in action. So this is a strategy we use. Like we started with the cells frozen, we freeze the 3D structure of the cell surface proteins with the chemical crosslinker, and again we biotinate them with again with the same strategy where we just oxidize the N-glycosylated protein, the cell surface proteins, and then install biotin here. And now we are not only just able to pull down the cell surface proteins, but we are also getting this crossing peptides. And this crossing peptide, what it tells us is if we get a stretch of amino acids which are close by in 3D space, and with that we get the rough 3D structure of the protein. And then we can compare the cancer versus normal cells, and then we can identify if there are some antigens which are conformationally selective. So for the first proof of concept, we applied it on acute myeloid leukemia. The very first reason there is no approved CAR T therapy against acute myeloid leukemia. So CAR T therapies at the moment works only for hematological malignancies. No approvals at the moment beyond that. So no approval for solid tumors. But even within hematological malignancies, it works only for multiple myeloma and basically B-cell malignancies. So for AML, there are no approved CAR Ts, and people all around the world is after it to find a CAR T or like come up with a CAR T approval against AML. So this is the disease that we want to target. So this was a workflow that we worked on. Like we used two different crosslinkers, DSSO, FOX, different kinds of high resolution mass spectrometers, like with MS3 capability, ion mobility capability, a series of fractionation techniques like SEC, high pH fractionation, IMEC enrichment, and all. We also had to develop this pipeline where we had to write our own softwares called like Wing, sudo MS3, Alpha Cross Excel. This Alpha Cross Excel we recently published in MCP. So we developed this whole pipeline, and then this is a paper, Alpha Cross Excel. We published it in recently in MCP, and using this tool, we identified this antigen called active integrin B2. So integrin B2 is normally present in T cells, in the normal T cells, granulocytes, and like it's broadly present in the hematopoietic cells. What we found is that these two domains, if you see here, the distance between these two domains is 38.9 angstrom. This is not something that we would otherwise see this. So this suggests that this is in active conformation. Sorry. Are the slides visible?
Yeah.
Yeah. So what this suggests is that these two domains are much closer to each other than what it appears to be here in the crystal structure. And that suggested that that this active integrin B2 is present in active conformation in AML, in acute myeloid leukemia cells, which is otherwise present in inactive conformation in other normal cells like granulocytes and T cells. So we validated this observation with different kinds of AML cell lines. We also validated this observation with the patient cells, and then finally what we did is we collaborated with Dave Sidhu's lab from University of Toronto, and then we were able to engineer antibodies against this active integrin B2 using phage display platform. So with our screening strategy, we initially found 10 hits, and then we funneled it down and finally identified one antibody which were amenable to be developed against in the CAR T format. And then we also had to screen like which antibody would work best again and would be specific to active integrin B2. So the way we identified is that in presence of manganese, the moment we add manganese, the integrins gets activated. So we tested the binding of this antibody in presence and absence of manganese, and then we found that 7065 is the antibody which specifically binds to active integrin B2, because here we see the moment we added this manganese, the signal is getting intensified. So that suggests that 7065 binds specifically to active integrin B2. We also had one more antibody called 7341, but 7065 appeared to be working better. We have a patent for both of these antibodies. We have a PCT patent, and we have also applied in the US. And this is how we engineer. We start with an antibody, take the variable heavy and the variable light chain, and we convert it into basically reconstruct it into a SCFV, signature variable fragment. And then all we have to do is just stick it onto the surface and just engineer the CAR-T backbone, and then we'll be able to display this SCFV on the surface of a T cells. Basically, just clone this SCFV in a CAR-T backbone. And this is how we make CAR-T. Start with T cells, and Tanveer explained it very well, that we just activate it with CD3, CD28 beads, and then expand them and engineer with something like lentivirus, and then we get this engineered T cells, and these engineered T cells are now better capable to act against cancer cells. So another problem that we got into, I don't know how much time do I have? Am I running out of time?
No, you can proceed.
Okay, so another problem, it's a very interesting problem that we ran into, is that integrin B2 is present in T cells. So when we activate the T cells, they get activated, and then we see like a lot of fratricide, like the T cells kills each other. So we had to knock out integrin B2 in order to manufacture the CAR-T. Otherwise, what we observed is that the CAR-T cells were killing each other while during the manufacturing process. And thanks to CRISPR, so with CRISPR, we were able to knock out integrin B2, and then we were able to almost like knock out CRISPR, like the modern with the modern technology, using the strategy of like RNP approach, we can almost like get rid of like 98%. The knockout efficiencies goes up to 98%, and now we were able to manufacture the T cells like without any fratricide. So this is how, then finally we were able to make this CAR-Ts, and when this is a challenge experiment in vitro, there's live imaging data. The CAR-Ts are labeled with green, and the tumor cells are labeled with red, basically mCherry and GFP. And what you see here is that the CAR-Ts were the winner, and the control plate, the tumor cells were the winner. We also ran this mice experiment, and we again see we also had the positive control here, CD33. So CD33 is the technical positive control of AML. This CD33 CAR-T does not work in clinic, because CD33 is also present in hematopoietic stem cells. So this is toxic. But it can be used as a technical positive control, and what we found is that our CAR-T was as good as CD33. And it was like there was drastic survival advantage compared to the control arm. We also had to address the toxicity part, and that is where single cell RNA sequencing turned out to be very helpful to us. So the single cell RNA-seq data suggests that the integrin B2 expression is very limited to hematopoietic cells, and so we don't care about toxicity elsewhere in the body. But even in the hematopoietic system, we know that it is present. We are targeting only the active conformation of integrin B2 and not the normal and not the inactive conformation. So we believe that we had a pretty good safety profile, which we also had to prove in vivo. So for that, we used this humanized immune system mice, where we reconstituted human immune system in mice we used a very specific strain of mice, NSGSGM3, and we injected human CD34 cells in mice, and then that's how we reconstituted human immune system in mice. We call it HIS mice, and we challenged these HIS mice with our CAR-Ts, and we see that the moment we use this CD33 CAR, there was drastic decline in hematopoietic stem cells of human origin. So we did take that with human CD45 antibody, and we see the drastic decline in human CD45 positive cells, whereas in our case, like the mice cohort treated with our CAR-Ts, they looks pretty much same as the control. We also did CBC profiling of those mice, and everything looks to to be normal without any toxicity. So just to summarize, we found a new class of cancer antigens which is protein conformation based and cannot be seen or detected with any other technologies other than at the moment we just use this structural proteomics technology to identify such conformation specific cancer antigens. We also use this technology to identify this active conformation integrin B2 to identify it as the AML specific antigen. And finally, we also developed a CAR-T therapy against AML targeting this antigens. With that, I would like to thank my postdoctoral mentor, Professor Arun Vita, UCSF. This is where I did this work, and thanks to my alma mater, NII. This is the lab. This is my current research group whom I'm working with. I'm thankful to all of them for their effort they're putting in. And I'm just presenting this work as they who work. And I work as assistant professor in Gujarat Biotechnology University. It's a new university established by government of Gujarat. So it's like dedicated to biotechnology. We have a 2 years MSc program and a PhD program. We have 5 different departments. So they are like specializations in animal biotechnology, environment biotechnology, industrial, medical, and plant. And all admissions are through Gate B for our MSc program. So with that, I would like to be open to taking questions at the end of this webinar.
Thank you. Thank you, Dr. Kamal, for a very nice presentation. Now I'd like to invite Dr. Rohini for her.
Yes. I think it's visible and I'm audible as well.
Yeah.
Okay. So thank you, Strand, for giving the opportunity to present the work. This is my postdoctoral work, which I will be mentioning. So currently in the current lab, we are doing single cell RNA sequencing, but I think, and we have 2 funded projects, one from ICMR and one from Gate Foundation. So maybe after an year, we will be having certain data to present. So this is the project which I did in my postdoctoral work, and it is mostly to identify the interactomes of RNA. So my talk will move from the fundamental RNA biology to the RNA interactomes, as well as my work, which mainly is also in the fundamental, using yeast as a model, asking the fundamental question. So just going to the history. So we should know that, sorry, I think for me it's not visible. So when Francis Crick proposed the concept of central dogma or the flow of information, I think at that time the mRNA was not identified. So he proposed the idea of DNA from mRNA to protein. But it would be amazed to know that mRNA was not discovered. It was only after the 2 years that in 1961, the concept means the mRNA was identified by Sidney Brenner and Francis Jacob. So, Francis Crick was the person who gave different concept like one gene, one enzyme hypothesis, one gene, one ribosome to one protein hypothesis, which was soon was discredited after the discovery of mRNA. So I think we should all know from the history how the mRNA was identified. And when he proposed the concept, the mRNA was not known, the visionary scientist, which was Francis Crick. So I think for the mRNA discovery, it was there was one physicist, which is George Gamhoff, and he tried to make a club which was known as RNA Tie Club. Here, in this club, there were 20 scientists he included, and this 20 scientists like Francis Crick, Sidney Brenner, Richard Feynman, and the main question was to identify how from gene to protein is being synthesized, and to understand to understand the mRNA. So this is the tie they used to wear. And you would be amazed to know that from the 20 scientists were named after the amino acid, and there were 4 honorary scientists, which was ATGC. And this scientist, you would be amazed to know that from 20 scientists, 7 won the Nobel Prize. So this is the history where how the mRNA discovery takes place, and it was Sidney Brenner and Francis Jacob, as I mentioned earlier, who discovered the mRNA because it was highly unstable molecule, and it was not very easy to identify. So if you come from mRNA discovery to mRNA therapeutics, now we are using mRNA as a therapy after Moderna, and Moderna has discovered the mRNA vaccine. So lot of progress has been made from the fundamental biology, because understanding the fundamental biology of the cell and identify very new molecules like siRNA, link RNA, mRNA, miRNA, which won the Nobel Prizes. And now we are in the era where mRNA or siRNA is used as a therapeutics like inclisiran or COVID vaccine, which we know so far. So this is the importance of having the fundamental, means to ask the right question, and how the fundamental insight can give, and how the fundamental insight can bring to the modern biology where we are. So if we talk about the RNA technology, there are various technology which has been developed. It is from RNA IP, which I will be mentioning later, to CellX and single cell RNA sequencing. Single cell RNA sequencing, I think Dr. Kamal and Dr. Tanveer has already mentioned in their presentation. So RNA IP is the main part in which you try to understand the RNA interactome, because it is also important to know how the RNA function inside the cells, because many viral proteins regulate by binding to the host RBPs. So it is important to understand the basic biology of the cells so as to make a target. Why it is important, because if you talk about RNA protein interactions, like RBPs, the how RNA is maintained in the cells, stability is mainly maintained by the RNA binding proteins, and also RNA protein interactions have other functions. Moreover, not only RNA protein interaction, it is also the RNA interactions which also takes place inside the cells. Whether you consider as stress granules or P bodies, there are quite dynamic cellular molecules which is present. So these all interactions are very important inside the cells, and how it regulates the cellular physiology. So, as I mentioned, the RNA protein interactions, if you consider from RBPs, which is important to maintain the RNA stability, or you talk about the risk complex, where in which the miRNA guided mechanism, which importantly regulate the gene expressions. These are the fundamental process in which the RNA or microRNA, or there are other forms of RNA which plays a very important role. Also, there are certain means the RNA protein interaction also play important roles in the modification of mRNA, which is the epigenetic modifications of mRNA, which also regulate the gene expression or gene status, because this epigenetic modification also regulate how the cells how the cells will express the mRNA. So, talking about the RNA interactions, we know that the basic mechanism like translation, transcription, splicing, black splicing, or the phase separation, these all are being guided by the RNA interaction. However, these interactions are not mostly studied, because I think there are not many technology which has been developed to study RNA interactions, or if we want to study specific RNA or mRNA interaction, this is not being studied mostly. However, RNA protein interaction, there are many techniques which are available. So the techniques which are currently available is like we used biotinylated RNA probes, in which you mix with the cell lysate and you pull down the proteins which is you pull down the proteins, and you do the mass spec to identify the associated proteins. So this is the most common method how you studied the RNA protein interaction. There are other methods also, if you see, like in vivo, if you want to see, you will cross-link or you will do formaldehyde fixation. You will fix the cells, you will lyse it, then you will use any antisense biotinylated oligos, and then you elute and identify the protein interactions. There are other methods like we have also developed, which is aptamer-based method, in which you add an aptamers. Either you integrate in the genome this aptamers after the stop codons and before the 3' UTR, so that you can pull down the mRNA, tagged mRNA, and see the associated proteins. So this is either you use IP or you do the proximately labeling like Apex or BioID. There is other which has been developed, which is Cas3 based approach, in which you use dead Cas13 biotin ligase, and this is specifically for the RNA of your interest. And then, like you used biotin and then streptavidin pull down, and you look after the associated proteins. So these are the methods which are being used. So what we developed was the aptamer-based RNA purifications, which we used to see both associated proteins as well as associated mRNAs. So since we were using yeast as a models, and we were asking certain fundamental questions, which I will be telling you later. But how we did was that we endogenously tagged the mRNA after the stop codon with an MS2 aptamers, and it was mostly mediated by the Kree-log system. Because what happened is, first, if you want to study the mRNA protein or mRNA interaction, ideal condition is that the 3' UTR should remain intact, because the 3' UTR plays a very important role in the mRNA localization as well as the function of mRNA. We mostly don't consider it because there are studies which tells that mRNA localization play a very important role for the cellular physiology, how the cell will behave. So if you want to, if you intend to study the in vivo system or how the mRNA behaves, actually, you need to tag the mRNA in such a way the UTR should remain in the close proximity or after the tag. So we used there is a tagging protocols which is available, and you engineer the yeast in such a way that the MS2 is tagged after the stop codon and before the 3' UTR. And this MS2 tag protein has the affinity to coat proteins, which is being fused with GFP. So the fusion with GFP, it helps for the localization of mRNA. So this is the advantage of using GFP, and then it is another again fused with streptavidin binding protein. So we can pull down after fixing the cells, we can pull down using streptavidin beads, and then we can either go for RNA isolation or protein isolation. I think the protocol optimization for us took a lot time, because here we always needed the proper control, so as to identify what are the associated mRNAs or what are the associated proteins which exist. And you can either go for RNA-seq or normal RT-PCR, which is feasible in any lab. So this is the advantage of this technique, that you don't need always to go for RNA sequencing. If you have certain known target and you want to see what is the interactions with your preferred mRNA, I think this technique has a lot of advantage. Why we went with this technique was we were asking one of the fundamental question in yeast. So we know that in prokaryote there is an operon kind of system which exists, like polycystronic mRNAs are formed, which helps in the coordinations of gene expressions. Means if you give a signal, there are polysystron which are exist, which is involved in the same signaling pathway. But if you see in the eukaryote, one genes for one signaling pathway is present in one chromosome, another is present in another chromosome. So the questions was, how does the eukaryote regulate the gene expression? Is it coordinated at the transcription levels? How the mRNA are being transcribed, or are they co-regulated together or not? So this was because we know that eukaryotic mRNAs are usually monocistronic. So, as I mentioned again, see, this is the coordination of mRNA from transcription to translation in prokaryote because it has polysystron, and in eukaryote it is monocistronic. So this was our question. Why we came to this question was when we were looking for the subset of mRNA localization, as I mentioned you earlier, that by tagging the mRNA with an aptamer, then this aptamer has an affinity to bind to the coat protein, which is fused with GFP. You can localize the mRNA inside the cells. And by using these methods, our previous works, means in the lab of my postdoctoral guide, when he did the previous work, he identified that many mRNAs are localized to the cortical ER because in yeast there are two types of ER. There is either cortical and as well as the nuclear ER. So there were many mRNAs which are localized to the cortical ER. Then there are mRNAs which are localized to the nuclear ER. Peroxisomal, the mRNAs are localized because it is always believed that mRNAs always goes to ER and then the translations start. But it is not the case because we have, and many other group has also identified that the mRNAs are localized to subcellular organelles, and this localization is very important for the translations of the protein in that specific organelle. So the question was, if these mRNAs are localized, means how these mRNAs goes there. Are they transported together or they transported individually? So this was the questions of our of my project. So what we did was we tagged the mRNAs which are localized in different subcellular organelles with the MS2 aptamer, and we pulled down these mRNAs with having the specific controls. And we found that certain mRNAs were enriched with the other mRNAs, like in yeast, there are mating mRNAs. And when we pulled down this subset of mRNA like SROS7, Exo70, and OM45, we found that they were enriched with the mating mRNAs. These were our RNA-seq data. But then, so we found that different mRNAs from different, and this was not that they are present in the single chromosomes. These mRNAs were present in different chromosomes, and they are coming together. So this was one of the initial result which we got, which gave us a hope that mRNAs also formed are complex together, and they transport from the nucleus to the cytoplasm. We validated with RT and as well as qRT-PCR that RNAs are pulled down together. But we wanted to validate it further. So what we did was we used two kind of approach. One was we tagged one of the mRNAs with MS2 aptamer, another with PP7. So this is visually we validated that these mRNAs are really co-localized together, and these two mRNAs are present in these are transcribed from two different chromosomes. And this validated that different mRNAs from different chromosomes multiplex into a single RNP particle. Now the question was how this mRNA comes, means what drives it. So we wanted to identify, because mRNAs, what is believed that mRNAs cannot go independently. They need to be associated with certain proteins. But when we looked for the proteins, when we pulled down with the streptavidin beads, and when we looked for the proteins, we found the presence of histone. But the histone is a protein which is present in the nucleus, not in the cytoplasm. But still, we wanted to see whether this histone has a role in the formation of these RNP particles or not. So what we did in yeast, there is a thing that we know that when you delete the histone, the cell will not survive. But in yeast, there are paralog of histone. So you have two copies. One is paralog. It is because of genomic duplication event. So there are two paralogs. So when you delete it, still the cell can survive normally. Okay. So when we deleted the histone and we looked for the formation of transferon, we found that there was less enrichment of the mRNA. So this validated that histone has certain role in the mRNA RNP particles formation. So this confirms that the histone has function. We also deleted whether it is only specific to histone H4. So we deleted other histones and saw the mating, because when you inhibit the formation of this transferon, we also identified that the cellular physiological process is also affected. Like since the mating mRNAs form RNP granules, this mating mRNAs has a physiological process of mating. So we checked the mating efficiency and we found that only histone H4 specificity was there. So this confirmed that histone H4 has a role. We also used AID Dagron systems in which you deplete the another histones for few hours till when the cell is alive. And we checked whether the transferon efficiency or the formation of RNP granule is reduced or not. And we found that when there is the depletion or when you add the oxygen, you found the cells in which there is no formations, there is no co-localization of two mRNAs. So this we not only did this, I'm not mentioning here, but we also did 3C experiments in which we identified that the mRNAs means the chromosomes are in very close proximity. The two different chromosomes are in very close proximity to each other. So there is a mechanism of co-transcription. And when you delete either histones, this proximity is also being reduced. Or either you use transcription inhibitors also, the process of the co-transcription process get reduced. So this was the validations. We not only validate only mating mRNAs, we also validated with other transferons like mitochondrials. Here I am showing about another, which is of heat shock proteins. So there was already one paper which gave us hope that the heat shock mRNAs, when you give heat shock response, the 3D conformation of the chromatin organization get changes and the genes come in the close proximity to each other. So we looked whether the mRNA complex also happens or not. So we did the same experiment. We tagged the gene with an MS2 aptamer. Like here we tagged HSP104 and we pull down the mRNA and we look for the other heat shock genes, whether they are found to be associated or not. And we identified that they form an RNP particles, which was validated both with IP experiments as well as with the fish fluorescence in situ hybridization experiment, that there is a really formation of transferon. So with this, we came up with a new concept of transferons, which is different from the operon system. And these transferons are like this, means whenever there is any stimulus, the genes which are involved in the same signaling pathway, they are co-transcribed, and then we now know the concept of co-translation. They are co-translated also together, and this lead to any physiological response. So this was our concept, which we proved in my postdoc. And right now we are studying ribosomal heterogeneity, as well as we are doing single cell RNA sequencing to identify different plague condition, as well as in the Gate Foundation project, which is mostly on heavy menstrual bleeding, where we are trying to understand the molecular mechanisms of different pathological conditions of the heavy menstrual bleeding. So this is my current lab, and I'm thankful to Weizmann Institute for doing such a beautiful piece of work in the basic biology, as well as the grant which I currently have. So thank you very much.
Thank you. Thank you so much, Dr. Nair. Okay, we will take the questions in the last. Yeah, let me have an. So I will basically talk about some of the strength point of view in this particular domain for the single cell site-seq and the CAR T cell therapy. So as nicely talked by Khushbu about the Strand Life Sciences. We are the 25-year-old spin-off from the Indian Institute of Science, Bangalore. We are based in the Bangalore, and we are in the different domains of the services, which includes the curations, bioinformatics, and the software development, as well as experimental and analysis. As a the today's more centered is the single cell and the spatials. I would like to inform that the Strand has a both the capabilities of the wet lab as well as the analysis domains. So in the wet lab, we basically performs the experiment. We have the 10x Chromium machine where we basically support the from the tissue to the library preparation and the sequencing, along with the in-depth analysis of the data. And this, in the single cell platform, we basically we supports all type of the single cell experiment that includes the single cell nuclear RNA sequencing, the single cell mRNA sequencing, the 5-valent single cell sequencing, which includes the mRNA along with the TCR, T cell receptor, and the B cell receptor sequencing. Also, the site-seq sequencing, where you can have the the surface protein quantification for each of the different each of the cells in, and also the single cell attack sequencing. We here have a well standardized pipeline for the analysis, which includes the very robust statistical method, as well as the different AIML based and the reference based methods to precisely annotate the different cell types associated with the sample or the tissue. In addition to the single cell, we also have an capabilities to support the spatial transcriptomics. It's a more the 10x based Visium HD, where which helps to not only look at a very close proximity with of the different the cell type, or we can say the microenvironment of the tissue. Also, we support in terms of the analysis of the another platforms for the spatial transcriptomics that includes the slide-seq, which is the CurioSeq, the NanoString Cosmex, and the GeoMax, and also the Morphis technology. So we can we previously handled many projects which is related to the single cell and the spatial biology. As nicely explained by both Dr. Tanveer and Dr. Kamal, I will basically move to the next slide because it's well explained how the CAR-T cell therapy works in the clinical perspective. So as we said, this new technology, which is basically a single cell based technology, which is called the site-seq. Here we have the two informations comes from each of the cells. One is definitely the mRNA count for each of the gene of individual cell, along with the surface protein quantification. So that can up to whatever the surface protein we want to look, we can explore for each of the cells. But when we go in more in the analysis, it is very crucial to understand the analysis and more precisely annotate, or before the annotations, your data should be more accurate, especially in the ADT, as the experimental process have three types of the. One is basically the native abundance of the surface protein. The more problematic is the estimation of the background and the ambient RNA. So in the overall analysis process, it is more important to precisely identify the threshold and selection of the counts. Here I will say counts because we have now the data in the forms of the counts for each of the different surface proteins we have considered. The counts which tells or which basically the true data over the background data. So there are the different methods in the literature which suggest or which explains how to analyze the data, how to identify the background and then identify the true signals or a true protein enrichment. We here at a Strand, we developed a pipeline. Why? Just one more thing that I said, that in the analysis, why it is more important, because sometimes when we look, or as you have seen here, there is only two bimodal peak here. You can see there's one that is represented as a background, and the right side is a true peak. But there are certain conditions what we have observed, that you have a more than two peaks, and sometimes there is a very minor fluctuations in the overall peak, and that somehow gives the uh, not correct data sets in terms of the estimation of the background as well as the true. So we basically here at a Strand, we developed in a pipeline which will take care of all the analysis, starting from the input FASTQ files till a complete analysis and a final report, where you can have the overall all the characteristics or estimation of the different proteins or the surface proteins, as well as your transcript of the interest. So we basically recently completed a project with a US-based pharma company, where we basically built in a pipeline that was for the CAR-T cell research, and the pipeline was designed to have starting from the FASTQ till the final report, and more importantly, it has a well characterized immunophenotyping modules, which will characterize the different cell populations associated with the data sets, as well as the amplification status of the CAR protein. It is very a scalable workflow because it's a Nextflow based, and it is compatible with the data set from the Fluent Biosciences as well as the SingleStop. So just not going more in details about the final outcome, I can just say that in a final outcome, you will have a report where you have a complete statistics of the cells, as well as not only the statistics, you can individually trace each and every cells of your experiment in terms of what are the different cell populations they are associated with, as well as a very nice representation of the charts, which tells how many cells are of which category. So this is what basically comes after once you have executed the pipeline, providing the details of the FASTQ files. You will have a final report, and the report basically have all these informations. Based on that, you can use these data sets for both the research purposes as well as for the clinical applications. Yeah, thank you. So this is you can reach if you have any thing related to both the experiment as well as the analysis. We are here at a Strand. We are happy to help you out in this. Now, I would request Khushbu to.
Yes, thank you, Dr. Pankaj. That was a very insightful presentation. I would request the audience to please put in their queries in the chat box. I can see that Dr. Arivu has answered one of them. There's an open question by Sanskar Rajput. Please feel free to add in your answers. How do we count for all statistical discrepancies resulting from the sparse matrix of genes containing zeros on most of the rows?
So we have basically a statistical method where we can definitely, when we go with the at an individual cell level, there are the many genes which is not expressed. And some of the genes have some of the experimental conditions you have with the noise. So the statistical methods we have implemented here, which take care of the date of basically these zeros and those which is we can say the noise in the data. And we basically filter out those cells when we go with the downstream analysis. So there are the statistical methods which we implemented here, and all those methods are well-established method.
Thank you, Dr. Pankaj. Anyone else would like to add to this?
Pankaj, I have a question.
Yes.
So the thing is, like, if you are doing 3 or 4 samples, and in one of the samples, like we recently did nuclei, single cell RNA sequencing. In some of the samples, the nuclei was less, and in some it was more. So finally, how do you normalize it?
So, so all are the single nuclei experiment.
Yes.
Okay. One approach is that first the normalization we do at an individual experiment level. That is the
Okay,
first
so
thing
that
we
is
do.
that.
Yeah, first thing,
Yeah.
because we cannot combine all and then
Mhm.
do the normalizations.
Mm.
Definitely, once we individually normalize, then we go for the integration.
Okay.
And
So
even in the integration, there are the many methods for the integration. So it's just not limited to the one. We individually look for all the 3 or 4 different integration method, and the method which basically justify more in terms of the biology, that we will proceed for the downstream analysis.
okay.
Okay, moving forward to the other question. For which solid tumors CAR-T therapy is available?
So let me, Khushbu, sorry, I have started traveling. There will be some background noise.
No
Am I
problem.
audible?
Yes,
Yeah,
sir.
yeah, yeah, Tanvi.
So for solid tumors, as of now, there is no approved CAR-T, but recent advancements have shown that CAR-T cells, at least for phase 1 and phase 2, to some extent have shown good response for glioblastoma, neuroblastoma, and China has now done phase 2 trial with the gastric tumor CAR-T cells using Claudin 18.2. For liver, again, GPC3 based CAR T cells, phase 1 has been done, but phase 2 is still pending. So, in short, no approved therapy for solid tumors, but multiple ongoing trials at phase 1 or phase 2 for many of these solid tumors.
Thank you, Dr. Tanvir. The other question is, how much NGS cost nowadays? What would be the cost to the system to acquire? I
Yeah,
think
I
Dr.
can take this.
Yes.
Yeah.
I mean, cost depends on, multiple things like what protocol, what panel, and things like that. So the sequencing cost has come down significantly, and it depends totally on what is your goal and what protocol has to be used and what kind of analysis. So there are a lot of parts to it. It depends on that.
Thank you, Arivu ma'am. There's one more question. I guess Dr. Tanvir could answer this. How Gen AI models may play a role in CAR-T cell therapy?
So AI has now transformed. I think the huge potential we see of AI is in healthcare and in energy sector. So because of the tremendous amount of data we have in CAR-T cells, AI is helping to develop these next generation models. We have AI model for CAR-T also. Our lab is also working on CAR-T agent, which shall be online in another couple of months. AI does help the screening which I presented. We screened almost 10,400 molecules. So it was possible because of the AI tools which we have developed. So it's not just the molecule design. AI also helps in the patient monitoring, clinical care kind of process development, preclinical study development. So in multiple domains. I think the data that was there earlier, where you needed to have multiple people working together, now with AI, it becomes so easy to analyze all the data at a single go. A huge application in this particular domain. I hope that addresses the question.
Thank you, Dr. Tanvir. Any more questions, audience, please, you can post it or raise your hands.
So, can I ask Khushbu question to
Sure,
Dr.
sir.
Kamal?
Sure, sure.
So, Dr. Kamal, it was such an
Yes.
excellent presentation. I am so happy to see the progress we have made. Such a beautiful work you are doing, particularly these
Thank
nano
you.
cards. We also try to develop something for AML, but I must congratulate you the way you have taken it forward. So
Thank
happy
you.
to see in India we are doing such nice work. So this nano
No, no,
cards
no,
you
no,
have
Selvi. This work was done in during my postdoctoral research. This
okay,
is
but you have
yeah.
continued working on that here in India as well.
Yeah, yeah. So now in India, I'm building a nanobody library from scratch because these nanobody libraries, they're all proprietary property of all the universities. Like they even if they share, it would come with a IP sharing, and you cannot like. So we at GPU, we are building this phase display library for antibody engineering, nanobody engineering in the each display formats. We're building these technologies here from scratch, and we would hold the IP.
No, I'm sure that you will be able to replicate the similar thing here because you have been doing
Yeah,
it
yeah.
yourself. So
Yes,
my
yes,
question was
yes.
about that CD 72
Sorry.
or the
72.
72.
Yeah.
So what is the status of that? Is the molecule patented by you? Can you take that to patients here in India? And how do you see taking that molecule forward?
So that is the work where I got exposed to CAR-T. So I'm not the inventor of that. I was like a second author in that paper. So the inventor was my postdoctoral mentor, Dr. Arun Vita, and the first author was Dr. Matthew Nix. So they, along with I think Andrew Cruz from Harvard University, they are the one who owns that patent. And I don't know whether they have already licensed that patent, but I know that they are at some stage in clinical trials at UCSF.
Thank you. Yes,
So
yes.
yeah,
No, but that is a very
if
exciting molecule. It looks really.
if yeah, if you're interested to do something about it in India, feel free to contact me. Like I can connect
Absolutely.
you with them. Yeah,
After this
yeah,
presentation, this
sure,
is over.
sure,
I'll contact you. Thank you
sure,
so.
sure, 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 sure. Yeah.
Any more questions from the audience? Okay, I think that summarizes our session today. I would like to extend a heartfelt thank you to all our experts who have taken out their time and attended this and given such wonderful presentations. Thank you, Dr. Kamal, Dr. Rohini,
Thank you.
and Dr. Tanvir for being here today and sharing your insights. And I extend a warm thank you to both our Strand experts, Dr. Aribu and Dr. Pankaj. Thank you so much. Dr. Pankaj, thank you for driving the session and introducing everyone and taking the session forward. And also, I thank the entire audience for joining us today and staying with us till the end and engaging with such beautiful questions at the end. Thank you, everyone, for making this scientific dialogues possible, and we look forward to more engagement. Thank you, everyone.
Thank you. Bye.
Thank
Bye.
you. Thank you,
Thank you.
Husbu.
Thanks,
Thank you.
Thank you.
Thank you, everyone.
Thank you.
Thank you, everyone.